Hello guys, I am back with another video on Fmge question. So this is the 23rd video of the Fmge question and I am very glad to announce it. So the classic example of missense mutation is thalassemia, sickle cell disease, sideroblastic anemia or hemochromatosis. So pause and guess the answer. This is a slight interesting question. So coming to the type of mutations, so we have point mutations. These are as the name suggests, point mutations. These are the changes in a single nucleotide base within the DNA sequence, and they are again divided into three categories. First is the silent mutation, a change in the DNA sequence that does not result in a change in the corresponding amino acid. Okay. So this type of mutation is not very harmful to our body as the desired amino acid is obtained. In missense mutation, what I mean that there is a change in the one amino acid, like in the sickle cell anemia. Okay, and non-sense mutation, a change in the DNA sequence that results in a premature termination of protein synthesis, causing truncated and other non-functional proteins. Okay. So we have other examples of mutations. So in insertion and deletion, example is a Tay-Sachs disease, duplication, Charcot-Marie-Tooth disease, inversions, hemophilia A, 22q11.2 disease, DiGeorge syndrome, translocation, chronic myeloid leukemia, chromosomal aberrations, Down syndrome, trisomy 21, and repeat expansion, Huntington's disease. Now let's talk about the options given in the question. Thalassemia. Thalassemia anemia due to decreased synthesis of globin chains of the hemoglobin. Okay. So as we all know that hemoglobin it consists of heme and globin. Okay. Now as the globin is less in the body, so it will cause a decreased hemoglobin, and this decreased hemoglobin will cause a microcytic anemia. Okay. and the patient with this with the thalassemia, they are resistant to the plasmodium falciparum malaria. Okay. And this is again divided into alpha and beta thalassemia based on decreased production of alpha or beta globin chains. Okay. So there are three types of hemoglobin found in the body. First is the adult hemoglobin. Adult hemoglobin consists of two alpha and two beta chains. HbA2 it consists of two alpha and two gamma chains, and fetal hemoglobin it consists of two alpha and two delta chains. Okay, uh, remember this thing. Now, option second, that is the sideroblastic anemia due to defective protoporphyrin synthesis. Okay. As you all know that the heme part of the hemoglobin it consists of iron and protoporphyrin rings. So in this anemia there is a decreased production of protoporphyrin rings. So it will cause a decreased heme, and consequently it will cause a decreased hemoglobin and will cause a microcytic anemia. Okay. So this slide shows the iron studies in the microcytic anemia, laboratory findings in the microcytic anemia. So pause and review the table. Now coming to the last option of the MCQ. So hemochromatosis. In this case, there is a excessive body iron causing deposition in tissue, hemosiderosis, and organ damage, hemochromatosis. Okay. There is a tissue damage is mediated by generation of free radicals. Okay, as we all know that the iron, it is a very toxic to our body, and free iron, it can cause production of a reactive oxygen species by the Fenton reaction. Okay, and 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 this hemochromatosis, it is a autosomal recessive defect in iron absorption primary or coronary transfusion secondary. Okay, guys. So our answer will be option B, sickle cell disease. Okay. So you need to know the causes of the disease, the clinical manifestation of the disease, and yeah, you're good to go. Thank you.